The purpose of our experiment was to look at biodiversity of mushrooms at different elevations to see if there would be a significant difference. Our hypothesis was that the higher the elevation, the lower the biodiversity of Basidia mycota found due to the lower resources available in comparison to lower elevations where more resources would be available. We went to 3 different locations: Reddish Knob, our highest elevation, Elkhorn Lake, our middle elevation, and the North River, our lowest elevation. We took the latitude, longitude, elevation, species of the mushrooms, and how many individuals of each species were found. We chose the plot by one member spinning around and another counting to 10 and saying stop. The direction the spinning member was facing is where we measured 10 meters and put our first flag. From that point, we measured 5 meters to the left and put another flag, and continued until we had a 5 meter by 5 meter square. We took a clipboard, a data sheet to record on, mushroom identification book, a hand lens, 4 flags, a measuring tape, and containers to bring some mushrooms back with us.
Overall, in our results, we found that the biodiversity of the fungi was at its highest at the lowest elevation observed, the North River. However, the second highest total of mushrooms was found to be at our highest elevation, Reddish Knob. Our middle elevation, Elkhorn Lake, showed the least fungi diversity. In the first graph, the number of species found at each location is shown. We found a total of 8 species at Reddish Knob, 7 at Elkhorn Lake, and 9 at the North River. In total, over all 9 sites, we found 24 different species of mushrooms. In our second graph, it shows the species found at the North River. We found 10 different species, and the total number of mushrooms found was 363. The average elevation at the North River that we collected samples at was 1,190 feet. Our second graph shows the species found at Reddish Knob. There were 8 different species found, with the total number of mushrooms being 71. The average elevation in which we collected was 4,363.3 feet. Our final graph shows the species found at Elkhorn Lake, with 7 different species found, and total number of mushrooms being 18. The average elevation in which we collected here was 2,180 feet.
Our data supports our hypothesis. The Shannon-Wiener index for each of the locations differ, which allows us to state that there is most likely a significant difference between the biodiversity and Basidium mycota at the 3 different locations. In addition, our evenness values differ, which allows us to conclude that between the Shannon-Wiener index value and the evenness value, there is a statistical difference. We did not find any of the same species in the highest and lowest locations. This was also true for a study done in Mount Fuji, Japan. In addition, a study in China found that there was a negative correlation between some fungal species and altitude. This also holds true for our experiment due to the fact that our highest elevation held the lowest Shannon-Wiener value and our lowest elevation held the highest Shannon-Wiener value.
Mushrooms are important to ecosystems of all types. They break down materials and reintroduce nutrients to the soil. Our experiment was important to ecology because we began looking into correlations between mushroom species tolerances. The ability for mushrooms to live in certain areas affects the type of ecosystems and nutrients that could be found there. Our data indicates that there is no significant difference between the biodiversity of mushrooms, and this is important to know for further research about the complexity, tolerances, and niches of